Okay, so 1.3, we will learn about the structure of the plasma membrane, so the most important organelle of the cell. So again, I don't want you to think that a cell membrane is it's just a simple boundary of a cell. In fact, this boundary has its complex structure. Once upon a time, we have several models that are now not accepted. And we have the Singer-Nicholson model of our membrane looking like a phospholipid bilayer. So how does a cell membrane look like? It will contain phospholipid molecules, cholesterol molecules, different kinds of proteins, and special type of protein attached to carbohydrates called glycoprotein. So let's look at the structure of a cell membrane. So for the test, make sure you can label the structure of the membrane. So you have these phospholipid molecules, okay? Then you have your cholesterol here, 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 here. Then you have different kinds of proteins. So those proteins that spans or crosses the membrane is called integral proteins, and proteins that are only found to one side of the membrane, you call it peripheral proteins. Now there are some special proteins that are attached to glucose molecules. So these are glucose molecules. You call this glycoprotein. So for each component of your plasma membrane, you need to discuss the functions of each. So your phospholipid, if I draw one phospholipid right here, that's your phosphate head and your hydrophilic tails. This is hydrophobic tails. This is hydrophilic and this is hydrophobic. Now, if you have that arrangement in your cell, it makes your cell membrane selectively permeable. So polar and non-polar molecules that needs to pass through the cell membrane have different types of transport mechanisms to be able to cross this charged particles of charged parts of your membrane. So your hydrophilic heads and your hydrophobic tails. So phospholipid has something to do with the permeability of your cell membrane. It does not allow any kinds of substances to pass in and out of the cell. Then you have your cholesterol. So cholesterol is in charge of maintaining the fluidity of the membrane. So it prevents the fatty acid tails from attaching to each other and freezing. It separates these fatty acid tails so it doesn't stick to each other. So it increases its fluidity. At the same time, I think it's mentioned on the report, your cholesterol prevents your membrane from being too solid, and it also prevents your membrane from being too fluid or permeable. Then you have proteins. As you've seen in the diagram, proteins can either be integral, it's integrated across the membrane, or peripheral, only to one side of the membrane, periphery, the side. These proteins, please memorize, serve different functions in the membrane. So they can serve functions for binding site, cell adhesion when cells need to stick to other cells. They can serve as channels or pumps for transporting different nutrients across the membrane, and also for communication, to communicate to other cells. And lastly, you have glycoprotein. This is not in the textbook, but it's good to know that there's also glycoprotein. So different cells will have their own distinct glycoprotein in their surface that distinguishes them from other cells. So phospholipid affects the permeability, cholesterol affects the fluidity, proteins affects the transport across the membrane, then these other functions as well. Now, this whole structure of your membrane, so this structure of the membrane is proposed by Singer-Nicholson, and they propose that the membrane is made up of two layers of phospholipid. So you have this layer, then the second layer of phospholipid, and you have different sizes of proteins, different structures of proteins embedded across the membrane or to the side of the membrane. And this is the accepted model of the plasma membrane. If you study the textbook, it's going to discuss the other models that were proposed prior to this accepted model. And let's talk about Gorter and Grendel. So what they were experimented on were red blood cells, and they found out that the phospholipid layer is twice as long as the size of the cell. So their conclusion was maybe the cell membrane are arranged into two layers. Therefore, the membrane is twice as long than the actual cell. So they were somewhat correct, but not completely correct yet. So there's two layers because the membrane is longer than the actual size of the cell. Now, Dobson and Danieli proposed the protein sandwich model. So let me borrow this from BioNinja. You have here the Dobson-Danieli model. So they propose that you have a layer of proteins here outside and here inside, sandwiching the phospholipid bilayer here. Now, it could have been correct. In fact, this model was accepted for more than 50 years. Now, make sure that you study the different experiments that disproved this model. So you have the freeze-etched experiment. So in the freeze-etched experiment, you freeze the cell membrane, and then you fracture the weak parts. So if it gets fractured here, because this is the weak part, what they discovered in experiment is inside here it's not smooth. There seems to be globules and patches of proteins. Now, how could that be if Dobson and Danieli says the proteins are here on the outside and here in the inner membrane, not inside? Therefore, this doesn't hold weight if, in fact, there's proteins here inside. Another experiment that disproved this was the structure of membrane proteins. Now, during the, what year was this? There's no year, I'm sorry. So advancement in biotechnology, we were able to harvest and isolate protein molecules. And what we discovered are proteins are not just one continuous layers. It can be made up of different shapes and forms. So it can be a globule, it can be a long chain. How can that be if in Dobson and Danieli's model, it's just one layer here and one layer here? During the experiment, they can have different forms. Another one is your fluorescent antibody tagging. So this is when you put fluorescence in the antibody so that when it gets into the membrane, it's gonna light up. So what the fluorescent marker is tagging are the proteins. So what happened in the experiment was there was fluorescence not only here on the outer layer, but even inside of your phospholipid bilayer. There seems to be fluorescence here. And it will only light up if it's attached to proteins. Therefore, proteins are not just found here as a layer, but embedded here across your plasma membrane. That's why we have this model now, the Singer-Nicholson model, where we have different shaped proteins embedded in the lipid or the phospholipid bilayer. Let's go back to our concept map. So to summarize, this is our accepted model. Wait, where is it? Where did it go? Sorry. So this is our accepted model of the cell membrane. Two layers with proteins, not here or here, but embedded across or at the periphery of the membrane. So make sure you can draw and or label the plasma membrane. Also memorize the different functions of your proteins, as well as know the purpose of that cholesterol, these cholesterol molecules, and also the structure of this phospholipid molecule, the phosphate heads and the hydrophobic tails. So, sorry, I forgot to mention. So these heads are hydrophilic, loves water. The tails are hydrophobic, they repel water. That property of a molecule is called amphipathic properties, meaning they have two different kinds of property. In this one molecule, this side loves water, but this side repels water. So that's all for the structure of the membrane.